Welcome to the webinar. I'll just share my screen and a few slides. I hope you can all see my my slide show coming up here. So I just want to jump into some of the data we have from previously, and show you a little bit about what we're talking about. So this is should be common to most of you. A picture of a mare and the reproductive tract, with all the different parts that goes into that. So you're probably all aware of that, especially the lower reproductive tract. So the vagina and the outer parts are naturally colonized with bacteria, but we also have different barriers to make sure that these normally occurring bacteria are not getting further into the reproductive tract and into the uterus. So the most sort of common barriers are the outer vulva. We have this vestibular junction, and also the most closed part is the cervix, which is on normal circumstances making sure that these bacteria are not entering the uterus and causing problems for the males to get pregnant. If we look a bit further into the uterine lining, this is a slide showing this quite complex mucosal lining of the uterus. And what you can also see here is the different parts where bacteria can hide. If we take a more schematic look of this, we see these caniculi that goes into the channels that goes into the mucus layer and makes a lot of nests or a lot of places where the bacteria can hide and colonize. We've looked a little bit further on this, and here we see these the glands where you also see a mass in the bottom of this. When we specifically go in and apply techniques that allows us to identify the different streptococci, for instance, you see that these small round balls here are actually streptococcal cells, which are colonizing these endometrial glands. Furthermore, we have also looked a bit further out. The black part here is the inside of the uterus. We have the mucosal lining here, so this is sort of what you usually will swap. But what you also see here is this very light belt basically below the mucosal surface. You may also see some blue dots, and you see a gland that is intersectioned here. We've highlighted that, and you can see that there's a lot of light here, especially the blue parts. This is also streptococci that are specifically colored with a coloring agent, showing that there are streptococci present in these uterine crypts. So not only on the surface, but also deep into the tissue, we find these bacteria. We've then also looked a bit further into how do the bacteria actually interact with the mucosal lining. And what you see on this picture is the streptococci here, in a nice chain, but also you see all this bit fluffy parts here. This is actually the equine cell that is enveloping the bacteria, and the bacteria are entering into the cells of the horse. You can see it more easily here. They simply communicate with the bacteria, communicate with the equine cells, and make them envelope the bacteria, so the bacteria can get inside the cells and hide from the immune system. So the bacteria are able to communicate with the equine cell and get taken up into inside the equine cell. Here you also see a nice picture, a long streptococcal chain making this invagination into the cell and enables them to be taken up. What happens then inside the cell is that the bacteria are forming a sort of a vacuole. You can see it outlined here. You can see the bacteria, the red bacteria, sitting inside, and this is a perfect place for them to stay. Sorry, this was not the plan to shift here. So the bacteria can hide inside the equine cell, and also if you treat the mares with antimicrobials, then they will not necessarily be up for treatment when they stay inside the cells. Another very important part is because normally Streptococci and other bacteria grow really well. They grow very fast. They will form large big colonies on agar plates. As you see right here, this is the typical streptococcal colonies coming up within 24 hours. But sometimes when the bacteria go dormant, as we say, or sleepy, then they form very small colonies that will only form within 3 to 4 days. And this is quite important to understand how a bacteriate is actually working, because these bacteria here are basically inactive. They grow very slowly. And what is important from that point is that most diagnostic methods will rely on bacterial growth, quite fierce growth, whereas most of the antibiotic treatment is also relying on inhibiting the metabolism of the bacteria. But these bacteria are hardly having any metabolism. They are very inactive, so they tolerate a lot of antibiotics, very high doses. So they are basically completely tolerant, and you are unable to treat them. So take home is that it's very difficult to diagnose because they are inactive, and it's extremely difficult to treat because, again, they are inactive and tolerant to the antimicrobials. So from this illustration here, it basically shows the whole thing. We have bacteria that are dormant or in a sleeping phase, and only when we treat them with bactivate, they will wake up and be visible as we usually see them, and also treatable with antimicrobials.